cloud is a little bit more kind of closer to being stretched along the line. Can you see that? Okay, so there's a better correlation here than there is here.
I'm sorry, a question.
Yeah.
What are the axes again? Like, what's 200, for example?
The activity during each.
Oh, right, I see.
I think this
What's
is.
the unit, though? I mean,
Yeah, no, that's actually a very good question. I think that what the activation here is activation. I'm not sure what the units are here. We'd actually have to look at the paper for that. But this is the activation. I think that each one of these dots is a voxel. Okay, so if you have a high correlation across multiple voxels between the activation for one repetition and another repetition, then all of these voxels are going to tend to, if it was exactly the same activation in every voxel and every line, then every voxel had exactly the same activation for the first repetition and fourth repetition, then all these things would lie perfectly on the straight lines. But yeah, no, that's an important. But actually, as to what the units are here, that's actually a good question. Because it's certainly not percentage signal change, that's for sure.
They say voxel, but I don't know how you'd have a negative 100.
Well, because voxels can have, you can have negative activation.
Oh, okay.
As well.
So those are the.
Yeah, this might be. Actually, it would be worth looking into methods to see exactly what these numbers are. That's a very good question. But this is essentially activation across a whole bunch of voxels.
Yeah.
It's my guess is it's probably something like percentage signal change times 100 or something like that. But that's a good question. But the key point, and I thank you for it's important to look at the details, and we should go and have a look at what the details are on this. But the key point here is that there's a strong correlation here and a weaker correlation here. Now that's just one example. Okay, so the crucial question is, how does this hold? Oops, how does this hold in general? And here's the average correlation for the remembered trials and forgotten trials. And you can see there's a higher correlation on average. And you can see these error bars are pretty small, so this is a significant difference. And this is the kind of actual data version of that kind of hypothetical correlation difference that we saw before. So in this case, brighter, more yellow, whitish colors correspond to higher correlation. So the remembered trials in general have higher correlation with each other than the forgotten trials. So that's kind of a nice result. I was saying that's really showing a direct connection between an important behavioral effect, namely, are you going to remember something or not, and neural similarity, suggesting that neural similarity is actually doing some work in the brain. It's actually at least related to something that matters for behavior. Now this does, however, raise a question, which is, we just saw in the Wagner et al. Study that you can get pretty good prediction of whether someone's going to remember something or forget it without looking at neural similarity at all, just by looking at activation intensity. You can get good encoding that seems to be strongly correlated just with having more intense activation just when the thing was presented. So you might say, well, if you can get better subsequent memory just from having more activation, maybe that's really what's driving this. Maybe it's not really, all the stuff about extra correlation at all. Maybe the extra correlation is just sort of going along for the ride and going hand in hand with extra activation intensity, but maybe it's the intensity that's really driving this. So then they did a nice control, and they said, okay, let's go in there. And some of the voxels really are going to be more active for the subsequently remembered trials. So let's get rid of all of those. And this is the nice thing about fMRI analysis. And for those of you, and we're going to be doing more of this, even if you're not doing this for your project, we're going to be doing more later in the semester in the computer practicals of actually going in, pulling out particular voxels, saying, find me all the voxels which show reduced activation in this condition, or which show greater activation in this condition. So what they did here is they said, let's find all the voxels with a fairly kind of liberal threshold, so we make sure we're getting all of them that are more active for the subsequently remembered trials. In other words, all the ones that might drive that kind of Wagner et al. Type effect. And let's just get rid of those. And let's get rid of those and then calculate the pattern similarities after having got rid of all that stuff. And then, will our effect still hold? And so that's kind of a nice control, and the answer is yes, it does still hold. So there's something extra happening with pattern similarity over and above what you get just from activation intensity. And that's kind of, that's often that type of question, not necessarily this exact question of, neural similarity versus pattern similarity, neural similarity versus activation intensity. But this question of, this thing that we care about, is that actually really giving us something extra? That's often a very, very important question to ask in any kind of analysis. And so that's actually, has exactly the same structure as the question that I was trying to emphasize before of, are we getting anything from the imaging over and above what we could get from behavior? Just because you spend a lot of time and effort collecting some type of data doesn't necessarily mean that it's buying you anything, right? One would hope that it is, but you have to actually test and see if it's buying you anything. And so this is an example of saying, all this calculation of pattern similarities, is that buying us anything? Comparing against behavior is a way of saying, having this multi-million-dollar fMRI scanner, is that buying us anything except for just having pretty brain pictures? And the answer is, well, yes, it can, but only if you kind of work carefully to get that. Okay, so that's one illustration of neural pattern similarity actually doing something, in this case being related to behavior. And for any of you are any of you interested in memory? Some of you are interested in memory. Some of you work on Alzheimer's and things like that, right? Yeah, including people whose projects I haven't replied to yet. Will be very soon. Sorry. So one of this is a very live question, right? People are trying to figure out ways of understanding what's going wrong when people forget stuff, right? In fact, this is kind of the biggest public health problem facing the developed world right now. That everyone's getting older and older and older. It's wonderful, lifespans are increasing. That's great, but lifespans are increasing, and people are losing their memory. Eventually, if you live long enough, it's kind of a depressing fact, but at present, if you live long enough, your chances of getting Alzheimer's just go up and up and up. Your chances of getting cancer, well, this is a separate question, go up and up and up. So living longer is great, but what we really want is longer healthy lives, not just longer lives. So people are losing their memories. What's going wrong there? So this is, one of the hypotheses is, well, if pattern similarity is driving successful memory, maybe one of the things that's going wrong is that the brain is somehow losing its ability to kind of make similar neural patterns. So these are some quite live questions. Okay, so that's one example. Now another example of showing that some effect actually is real or has some implications beyond just kind of being something you can measure. Well, one way, a nice good way is trying to tie it to behavior. Another way is saying, can we find some kind of regularity across people? Does this find something? Does this relate to something that's kind of out there in the world? That's not just in one person's head, but which actually seems to be in multiple people's heads. So we'll have a look at that in a second. But before, anyone have any questions about these kind of memory types? So we already had a good question about what exactly is being plotted on these axes here, and I'll actually have to look more into the methods section of the paper to see exactly what they're plotting there.
I had a question that was kind of confusing me, and I don't know how you apply this to using MVPA, but
okay.
There's sort of the phenomenon of repetition suppression, right? And so, which is like in the degree to which a voxel might be specialized for something. More repetition of the stimulus, you find that the bold signal will decrease, right?
Right.
And so it's somewhat surprising that they find the opposite, that you see it more and you find an increase signal.
Very interesting point. So just to kind of unpack this question a little bit for everyone. So one thing that's been found in general, not just in fMRI, but as a kind of general property of neurons, is that if you just kind of make a neuron fire again and again and again, it's going to get tired, kind of just like us, right? Okay. So if you say you've got a neuron that likes a particular face, right? If you show that face, the exact same face 10 times in a row, then neuron's going to start off saying, "Oh yeah, it's my favorite face. It's my preferred stimulus." They're literally called the preferred stimulus. It's going to fire a lot, and then gradually less and less. It might be to do with it, having depleted levels of neurotransmitter or something like that. So it's in general been found that if you present the same stimulus over and over, the neurons that are sensitive to it will over time respond less, and that's called repetition suppression. That's actually, by the way, a nice method for trying to figure out what kind of things count as the same or different for a population of neurons. This is a whole method in fMRI called adaptation fMRI, which we haven't talked about so far, but I'll just kind of mention briefly. Suppose you think that, say, a neuron can tell the difference, or a brain area can tell the difference, say, between faces seen at different viewpoints. Okay. Well, one way of doing that is to say, okay, I'm going to show face of a particular viewpoint lots and lots of times, and the activation is going to go down, 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 down. And then if I change the viewpoint of the face that it's seeing, all of a sudden I'm going to activate a different population of neurons, and those old neurons, which aren't tired because those are fresh, and then I'll get more activation again. So you get a release from adaptation. It's called release from habituation, so the signal will go up again. And that will be a signal that, yes, this population of neurons altogether really can tell the difference between faces at different angles. Whereas if the neurons just respond to faces in general, they don't care what angle, viewpoint you're seeing it from. When you change the angle of the viewpoint of the face, they'll just keep on getting tired because, it's just more faces. Okay, so this is actually a kind of nice method for looking at cell activity. Anyway, to get back to Frank's question. So if you're observing neural repetition, you see all the time. It's a very, very common thing. So if the neurons are responding less and less and less, then how could you, how could this be consistent with getting greater pattern similarity? Well, this is one of the this actually raises, this nicely highlights one of the things about, one of the key points about pattern similarity that distinguishes it from activation. So you can have two patterns be perfectly correlated with each other, even if they have different levels of intensity, right? So for instance, I sometimes like to use this analogy of like a black and white photo, right? If I take the exact same photo and I just kind of turn the contrast down, it's still the same photo, right? It's still showing the same thing. And if I take the correlation between the high contrast version of the photo and the dimmed down version of the photo, I'll get very, very high correlation because it's, in fact, I'll get correlation of 1 because it's exactly the same photo. But the intensity is going down, down, down. Okay? So I'm not, I wouldn't be too surprised if they even looked at that in one of the supplemental figures because it's a very, very good question. But what's quite likely here is that what you're getting is actually the multiple presentations of the face. So you're getting reduced activation, but the same kind of pattern, just like you're having kind of fading out versions.
Preserved, the variance is
exactly
maintained.
So the pattern across the voxels can be maintained, even though all of them might be going down a bit. I mean, one complicating factor here is that actually different faces were getting interleaved with each other. So in the presentation, it's not the case that they were just seeing the, so it's not the case that they were seeing, face 1, face 1, face 1, face 1, and then face 2, face 2. So they were all being interwoven. So you might see face 1, and then you might see face 2, and then you might see face 1 again, right? So when you have lots of different types of stimuli being interwoven like that, interleaved, that can actually affect the habituation over time because the face 1 sensitive neurons get a little bit of time to recover. But that's actually a very, very good point. And if those of you are interested, you can go and download the supplemental information and you can see. I wouldn't be the least bit surprised if that's actually something that they looked at, because that's a key question. Any other? Yes, Ruan.
So for the MVPA, so if I get some like learning process, read some paper about the learning process. If you say, let's say, for MT, you try to learn some like direction or like let's say before the learning, then you have no performance very bad and you cannot do like decode sort of like information. You cannot classify sort of two patterns.
Right.
For example, after learning, you get really good at this task. Now, if you scan MT or V1, just for example, Frank-Tom paper,
right,
you can get like sort of like different patterns.
Right. Right.
But the question is, if this sort of similarity or does how, if you think about like different voxel, the more similarity means like more, maybe I'm wrong, like less variance. If you think about like sort of a correlation, that means after learning or something after attention or after adaptation, those, if you compute from the computational like idea, like the entropy, the information you carry, you like transmit should be less. If you think about like noise correlation, if you think about like the spike, like some argue like if you like the more correlate with each other, like between different neurons or different voxels, the same idea like the spike, like the entropy, how much information you carry, should be less. So I'm wondering why this should be the case.
I think that's an interesting question. I think there's quite a few things going on in that question, and you and I should chat about a bit more about this later. But I think that one, that's an interesting question. I have to think about that, and we'll have to talk about that more. But I think one thing that's worth bearing in mind is that, in the last class we were talking a little bit about how there's this kind of technical measure of information in terms of, like how much uncertainty is there in the distribution, how compressible something is. That's not necessarily the same thing as the kind of information that we kind of intuitively think about, or even just non-intuitive, that actually kind of information about the world. So, I gave this example of, I could increase the amount of information in, that if I download the entire text of Wikipedia, I could increase the amount of information in that kind of entropy type sense by turning every single character into a random letter. Okay, right? In terms of like ability to like extract useful information in terms of, I want to learn how to, build a house or something, so I go to the Wikipedia page on house construction, right? I've completely destroyed that information, right? Which is kind of what we care about. But in terms of how compressible is this text file, now it's like way less compressible. So in that sense, it has more information. So I think that's kind of, I'm not sure if that actually necessarily gets to the key point of this particular question. But I think it's worth bearing in mind these two things aren't necessarily the same thing. I'd be interested to chat with you more about that afterwards. But I think one of the things that this highlights is that nobody actually knows what is the key information in the brain and how is it being stored. This is one of the reasons that neuroscience, cognitive neuroscience, is kind of an exciting but also a difficult area. That, if we could just say, well, there's this stuff out there in the brain and it's encoded like this, so let's go find it, right? Then we'd be all set. But we are in no way even close to being able to do that. We can say, well, we have this hypothesis that maybe information might be being stored in this way, which we have an extremely imperfect, indirect way of sort of measuring, kind of. So let's go and see if we can measure that thing, and let's see if we can pull some information out. And this is information more in the sense of, news you can use, more in the sense of like the content of the Wikipedia page rather than like how compressible it is. So in this case, it's saying, okay, we can actually go in there, look at the pattern similarity of these neural activation patterns and say, haha, when there's greater pattern similarity, somebody's more likely to subsequently remember this. Now that doesn't mean, it definitely doesn't mean that the brain is somehow, kind of, there's some part of the brain that's looking at some other part of the brain that's kind of subdividing it into voxels, taking the average activation across those voxels, correlating it, and deciding whether to remember or not. Okay, certainly not. No way. But maybe there is some distributed pattern of actual neural activation that we're sort of indirectly picking up with these voxels, and maybe we're kind of tapping into that stream. So, but as to the question of, how is the stuff really, really encoded in there, nobody knows. A lot of people trying to find out. And so I think your question maybe kind of also illustrates the fact that the really fundamental question of, how is stuff really being encoded in there, we still don't know. And that's you know, that means that we're gonna run out of questions to ask you, but it also means that we're kind of groping around in a very dark room. But not a completely dark room, right? There's some kind of, you know, glimmers of light in there, but and that's kind of the state of the field in neuroscience right now. But that's a very, very interesting question. I'd love to chat with you more about that. But I would definitely, you know, highlight that when you know the term information is used, it can mean this kind of slightly non-intuitive sense, in the sense in which, you know, you increase the amount of information in a text file by turning all the letters into random stuff, and you got to be a bit careful about that. Okay, so here's a different way of looking at neural similarity, and this is actually, you know, the paper, one of the papers I did put in the readings. It was me kind of indulging myself a bit by putting one of my papers, but hopefully, you know, I think this actually raises some interesting questions. And this, again, also highlights that there's lots and lots of things that we don't understand. This is just, you know, starting to get towards some understanding of some of these questions. Okay, so so here we are, you know, representation of information, right? Nobody knows exactly what representation means in the context of the brain, and nobody knows exactly what information means in the context of the brain. But we can at least get some handholds on it. So we'd be getting a handhold if we can find something that's true not just in one person's head, but it's true across multiple people's heads. Okay? And this is actually not that easy to do. There's all kinds of reasons why it's difficult to find things that are in common across different people's brains. And a big reason is because everybody's brain is a little bit different. So remember we were talking about in the pre-processing step in one of the computer practicals that, you know, one of the stages is to try and squish everyone's brain onto a standard template. Okay? So that involves a couple of things. It partly involves rescaling, because some people, you know, just have bigger heads, smaller heads. But also it involves a kind of non-linear squishing, because, you know, some people, some parts are a little bit, you know, scaled out, a little bit bigger, a bit smaller than others. And that's not the only sense in which people's brains are different. So especially when you get to kind of higher level areas, their locations, their exact locations in the brain can vary quite a lot. So even if you can perfectly align every brain to the template, that still doesn't mean that regions that are specialized for a particular thing will be in the same spot in that template in every person's brain, especially not the high level regions. So, you know, everybody's primary visual cortex is right at the back of their head, and there's a particular sulcus, this kind of big one here called the calcarine sulcus, that's always smack bang right in the middle of V1. Okay? So if you want to just anatomically identify where is somebody's primary visual cortex, you can do that pretty well just looking at raw anatomy. Okay? But for higher level areas, that isn't going to work. So here's an illustration. This is from a paper by Georg Langs, but there's a lot of papers showing this, of where, in this case, a couple of language areas. So, so there's two main, you know, what you hear all this stuff about left brain, right brain, but pretty much the only aspect of that that's really true is that on the whole, language is mostly on the left. Not for everybody, especially not if they're left-handed, but language does tend to be on the left. And there's two key language areas, as I expect you all know. Broca's area, which is kind of on the left frontal, kind of around here, and Wernicke's area, which is kind of left posterior, kind of around here. And so this is in green and blue, is kind of Broca's and Wernicke's area in different people. And what you can see is they're all kind of sort of in the same general place. Like, you know, Wernicke's is always back here somewhere, and Broca's is always up here somewhere. But they're not in exactly the same spot, especially if you kind of try and align them with, you know, particular folds. So the folds are called sulci, and the bumps are called gyri. Okay, so so you know, here's Broca's is kind of a little bit further forward, and this person is kind of a little bit higher up in this person. So there's variability across areas, and that's at a coarse level. That's at the level of entire regions. But what if we look at a finer scale? Well, a finer scale, you know, when you get down to the level of, you know, the individual little folds or even the neurons within these folds, they grow in their own way within each person. Everybody's fine-scale neural structure is different from everybody else's fine-scale neural structure, and that's one of the key things that makes us unique as individuals. And there's lots of things that make us unique, but that's one of them. And so a kind of metaphor that may actually be a little bit more than a metaphor is that you can think of them as a little bit like fingerprints on the hand. Okay, so if you stick your hand up and I stick my hand up, they're going to line up. My hand might not be the same size as yours, but it's going to have on the whole 4 fingers and the thumb, so they kind of line up. But if you say, well, does that mean my fingerprints will line up with your fingerprints? No, okay, because at this fine scale, they're different. And it's kind of like that with brains too. So that, we've all got a temporal cortex here and a frontal cortex here and parietal cortex here, unless you've had some kind of horrible brain injury. But the exact pattern of folds, the exact pattern of neural connections is more like the fingerprints, which are kind of unique and idiosyncratic to individuals. And you can think of the activation that's made by particular stimulus. Say we all look at a chair, right? We all look at the same chair. Okay? Well, that's going to produce, in all of our brains, that's going to produce activation more or less in some particular regions of ventral temporal cortex, probably fairly distributed regions of ventral temporal cortex. But the exact pattern that the chair makes in my brain and the exact pattern of activation it makes in your brain is not going to be exactly the same. You can think of that as the neural fingerprint of a chair made in my brain and your brain, and they will be kind of idiosyncratic to us, just like the fingerprints in our fingers. So this is a sense in which this might go a little bit more, be more than a metaphor. And in fact, people have looked at that, looking to see whether you can look at the actual activation patterns in one person's head and use that to find similar, exact same patterns across the same set of voxels, when you line people up to the same template in other people's heads. And the answer is that often you can't very well. So here's a nice quote from this paper by Shinkareva, Mitchell and colleagues. It says, critical diagnostic portion of the neural representation of category as an example, that's what I'm looking at here, is still idiosyncratic to individual participants. Okay, so my neural fingerprints are different from your neural fingerprints. So there's been different attempts to try and look at this. Now, one very important attempt, which I encourage you to go look at, but I didn't post one of the readings, is this paper by Jim Haxby and colleagues looking at what they call hyperalignment. You'll recall that Haxby was the person who kind of kicked off this entire area of pattern-based neural decoding in his 2001 science paper. So very, very important work. And recently he's been looking at this question of what's common across different people, and what they've been trying to do is kind of take the voxel activation pattern in one subject and the voxel activation pattern in another subject. This is very high dimensional, thousands of voxels, and try and find some kind of high dimensional mapping of one to another. That's what they call hyperalignment. So that's one approach. But another approach is to say, well, maybe that voxel space activation is not actually capturing what we want. Maybe we need to somehow abstract away from that. Well, what are we going to abstract towards? Well, how about if we look at similarity relations between the activation patterns? So not the activation patterns themselves, but the similarity between them. In this case, it's measured simply by spatial correlation. And so the hypothesis is that this actually might reveal some population-level regularities. And a good test of whether you've actually found population-level regularities is whether you can decode one person's neural activation using information from other people's brains. Okay, so if you can do that, that's kind of like using one person as the testing set and the other people as the training set. If you can do that, then you've found some regularity that spans across all of those people. So then this raises the question, okay, well, we want to do some kind of decoding in just using similarities. So how are we going to do that? Because remember, typically what you do is you take a neural activation pattern, which is in the voxel space, it's a bunch of activation across voxels, and you throw it into some kind of classifier, and the classifier says, okay, given this pattern across a whole bunch of voxels, I think that it was a face, or I think it was a chair, or I think it was a house. Okay? But if you're going to work not on the actual activations, but on the similarities between those activations, then you're dealing with a completely different thing. You're dealing with one of these similarity matrices. You're not dealing with pattern across thousands of voxels. So suppose we have a bunch of similarities and we want to try and decode them, which basically means we don't know what the true labels are, but we want to figure out what the labels are. That's essentially what decoding is, attaching labels to things that we don't already know. So here's an example. Okay, so suppose I said that A, B, and C here are 3 different cities, and the positions, the distances between them, which is similarities in this case, correspond to the actual geographical distances between cities. But I'm not going to tell you which label corresponds to which. I'm just going to say one of these is New York, one of these is San Diego, and one of them is Boston. So can anyone say which one's which? This is not a trick question, by the way. Yeah, Adam, yeah.
Well, San Diego's C, New York's B, and Boston's A.
Exactly, exactly. Okay, now the question is, how did you do that? How did you figure that out?
My knowledge of geography.
Exactly. Okay, but here's exactly. Okay, so here's a kind of here's an approach which would work. Okay, which is almost certainly not how you did it, but which is a kind of suppose you were going to take a more kind of dumb brute force approach. You could say, okay, I got these three labels A, B, and C, and I got these three cities, Boston, San Diego, New York. I'm just going to try all the possible labelings. Okay, there's actually 6 different ways of assigning these different labels to these different cities. I'm just going to try all of them, and for each one of them, I'm going to see, well, how well does that actually correspond to the actual distances between the cities? So I might say, okay, let's try, let's say A is San Diego, and B is Boston, and C is New York. Okay, well, with that labeling, then San Diego should be close to Boston. Oh, well, that doesn't work. Okay, so I guess that labeling's wrong. Okay, so you could literally just try all the different labels and say, given this labeling, how well does that correspond to the actual set of similarities, and then just pick whichever one gives you the best match. Okay, so it's not it's a very kind of brute force approach because you literally are just trying everything. But as long as you don't have too many different labels, then it could actually work okay. So I applied this on the Haxby data, which has 8 different categories. There's like faces, houses, chairs, cats, scissors, bottles, scrambled pictures, stuff like that. Okay, and so the number of different possible labelings is actually 8 factorial, which is like 8 times 7 times 6 times 5 times 4, which is like 40,000 something. Okay, so it's quite a lot, but if you just want some computer to churn through it, it can do it in a few seconds. Okay, so this is just one of these similarity matrices where you've got the 8 conditions, but we've taken away the labels now, and we've said, okay, one of these is cats, one of them's houses, one of them's bottles. But I'm not going to tell you that. I'm just going to, just as I called those cities A, B, C, I'm going to call these A, B, C, D, E, F, G, H. And then you say, okay, so I've got my training subjects, which is all of the, in this case, the 6 subjects. So I'm going to take 5 of them, and I say I know the labels for those guys. Those are my training. And I'm going to take my test subject, and I'm going to take away the labels for that. And I'm going to say, if neural similarity really is capturing a true regularity across the different subjects, then if I just look at the neural similarities and I try and match the neural similarities in my testing subject, who I've thrown away the labels from, if I try and match the neural similarities for that person as closely as possible as to the neural similarities of all the other people who are my training subjects, I do know their labels, if the neural similarity really is capturing what's common across these different people, then trying to match neural similarities should end up giving me the right labels. In other words, I should be able to successfully decode across people. So that's what you do here. So this is the average similarity matrix from all the other people, where we actually do know the labels. So it's like shoes, scrambled pictures. I don't know if you can see this very well. Scissors, houses, faces, chairs, cats, bottles. Okay? And then over here we've got a test subject, and we just try all 40,000 different labels, okay, which just takes a few seconds on a computer. And you say, okay, I've got A, B, C, D, and H. Well, maybe, let's try this labeling, where A is scrambled picture, B is shoes, C is houses, etc. Well, that would produce this similarity matrix. Let's see how good that's similar. Let's see how well that correlates with the actual average from the actual people. And then we just literally look at the correlation between them. And then you say, okay, try that for every single possible labeling, and just pick whichever one is the best. Okay, whichever one gives the best match, the highest correlation in this case. So if neural similarity space is actually really capturing what makes different people's representations alike, then jiggling around the labels to give the best match of the neural similarity matrices should end up giving us the right labels most of the time. So you've got these 8 different categories. So you've got 6 subjects in 8 different categories, so you have 48 different labelings altogether. And it actually turns out to give you the right answer 44 of the time, 44 of the 48 times. So it actually does pretty well. And you know, because there's 8 different labels, then the chance performance would just be 1 in 8, which is 12.5% correct. So what that says is that in this particular instance, neural similarity really is actually, the similarity matrices really are quite similar across different people, such that if you take one person's neural activation, you say, I don't know anything about what this person saw, except that, you know, some of the, you know, there's 8 conditions, and of that bag of 8 conditions, that, you know, one of them was faces, one of them was chairs, one of them was bottles, but I don't know which one's which. You can say, okay, I can actually assign the right labels to those conditions in this person's head just by looking at what the labels are in the conditions in other people's heads, but not by actually trying to match actual neural fingerprints, actual neural activation patterns, but by trying to match neural similarities instead. So that suggests a neural similarity. So we've seen from kind of 2 different angles here the idea that neural similarity is really kind of capturing something kind of real. One, that it connects with behavior, in this case kind of subsequent memory. This is what I looked at in the first half, and this one that is capturing some regularity across people. Yeah?
So if you think about this decoding process, even though like the Franklin's paper is just sort of like 2 orientation, but it's all, for me, it's sort of like 2 binary like classification.
Right.
Is there any possible like to decode some continuous variable?
Yes.
Like.
Yes, people look at that as well. That's a very, very good question. And in fact, actually, Tommy, you were asking this question once as well. There's actually a nice review paper about this. So Tommy actually asked me this question a while ago. Before I did the whole Piazza thing, I tried setting up this like Facebook discussion group, and very few people, thank you, who actually posted a question that was Tommy. And he said, well, the question he posted was, have people looked at trying to decode continuous variables, not just, you know, like kind of discrete things, like is it this or this, or you know, and this, that, like often it's between 2 things. In this case, it's between 8 things. And the answer is yes. And I will actually pull up, I found like a nice review paper on that, and I'll pull that out from my email and I'll post it on, you know, I'll send it, I'll post it to Piazza or something. And essentially the way in which people do that is, so people look at classification, where essentially you want to, you know, make some mapping and then apply a threshold and say, okay, if it's above threshold, I'm going to say it's class 1. If it's below threshold, it's class -1 or something like that. And people also do regression, where you basically do the same thing, but you don't do the threshold bit. Okay? Where you say, okay, I'm going to find some mapping of these neural activation patterns to some continuous variable, and I'm not just going to chop it up. I'm going to say, you know, let's see how well we can actually get to this continuous variable. And so there's all kinds of different ways in which you can do that. You can do standard regression. You can do what's called SVM regression. You can do all kinds of things. And essentially, they allow you to try and to map to a continuous variable. And there's all kinds of continuous variables. The variable might be something like, one example is, you know, how old is somebody, right? There's actually papers that will take different aspects of neuroanatomy and see, can we predict someone's age from their brain? Or people have also looked at kind of functional connectivity, can we do that? Or it might be a kind of health measure, you know, like, you know, it's not just the case that somebody, you know, has Alzheimer's or doesn't have Alzheimer's. They have different degrees of impairment, right? So yeah, people do look at that, and I will pull out that paper and look at it, but and send it to people. So yeah, I've mostly been discussing decoding and mapping onto kind of, you know, discrete outcomes, so this or that, and that is actually remains the case that that's often what people look at. But the question of continuous variables is also very interesting, important. People look at that too. It's a good question. Anyway, if any of you, this is a kind of last quick point, if any of you happen to be interested in philosophy, which I know some of you are, it turns out there's a very good philosopher called Paul Churchland who actually asked the exact same question, but in a different way, in purely conceptual grounds. He's got this nice paper called Conceptual Similarity Across Sensory and Neural Diversity. that I published in 1998. And he basically asked the same question: given that one brain is different from another person's brain, how can these different brains have the same concept, right? How can they be representing the same thing, given that the neural substrate is definitely going to be different? And so what he suggested was, well, maybe what you need to do is you need to abstract away from the actual neural patterns, because they're not going to be the same, because everyone has different neurons, and instead look at the similarity relations between these different concepts. And the thing that makes one person's set of concepts the same as another person's set of concepts is that they have the same similarity relations between them. So anyway, so I was actually just kind of struck that this approach, on purely conceptual grounds for philosophy, actually turned out to converge to a very, very similar solution to an approach that came from a completely different direction from fMRI. So, you know, sometimes if you kind of find that two different approaches that come from very different places end up converging on a single point, that kind of suggests that they might both be being guided by something real. So anyway, so those are some different ways of seeing that neural similarity might actually have something real going to it. So next week is spring break. So please enjoy yourselves, you know, on the beaches of Cancun or wherever you're all going to be going. And I'm going to finish replying to everyone's project emails. Sorry, and B, I'll send out essentially this and this saying, okay, let's take the next step. And maybe next week will be a good time for people to kind of catch their breath a bit. And also, you know, not as well as just replying to emails, I don't know, we can discuss things by email, but we can also meet in person and, you know, and kind of start pushing these projects along a little bit. And I've been kind of a roadblock in that so far, so that roadblock is being removed. Right. Have a good spring break, everybody. See you in a week and a half, I guess.
Did Andy Connolly do his postdoc gymnastics?
Yeah, he's still doing postdoc gymnastics.
I think I've seen him present those data before.
Oh, okay. And he did also some very nice work on decoding different animal categories.
Yeah, maybe that's what I was thinking.
Yeah.
He has a paper
that came out in January.
This was in 2010 when I saw him do this. So
oh, okay. Well, he might have been, his J neuroscience paper, I think came out in 2012, but he might have been presenting it, you know, like before it actually came out. Yeah, he's a good guy. Which conference was this?
This was in Rovereto.
Yeah.
Oh, okay.
Like,
Nice.
He was, I think he had a poster, but he won he had a
go up to
travel award.
vendors,
Oh, okay.
telling
So
them
he was
here's my
one of the
invention, like go up
Great.
to marketers saying, market